So the reason Hubel and Wiesel decided to do the research the way they did it was because they wanted to understand the pathway from the stimulation of a receptive field on the retina all the way into the brain. So one of the things you have to remember is that when we say receptive field, we mean a spot on the retina. Okay, on the retina, because the retina, in the case of vision, the retina is where the receptors are located. So that's where the receptive fields are located, is on the retina. And basically what they were trying to do was to stimulate a receptive field, one receptive field on the retina, and try to understand what wiring was activated, what neural wiring was activated going from the receptive field on the retina all the way back into the brain. This is a work for which they were awarded the Nobel Prize also. It's very important work. So the model that they used is illustrated here on figure 324 out of your book on page 63. Usually anesthetized cats or monkeys were used, and they would place the animals in an apparatus so that their eyes were focused on a screen in front of them, and they would use a lens to focus light specifically onto different spots on the retina. So in this example here, they would be looking at a visual field that would have three spots on it, A, B, and C, and of course the spots would be projected onto the back of the retina in the corresponding fashion as they're illustrated here. And so let's say they would light up spot C. They would light up the spot C, and then they would go looking for a nerve fiber in the optic nerve that was being activated by the receptive field at that spot C. And from there they would trace back the signal as it went from the retina all the way back into the brain. This picture here shows in the human brain the circuit that we're talking about. The image at the bottom here, basically this image is if you were to take and slice off the top of the head, like going right straight through here like that, and then remove the top and then look down. This is a cartoon of what you would see. You have the eyes, of course, right here facing forward. Information travels in the optic nerve to the optic chiasm, which is a crossover point, right? So half the information in one visual field stays on this side of the brain. The other half of the information travels to the other side of the brain. And then the information goes from the optic chiasm to the lateral geniculate nucleus, and also a bit of it goes off to the superior colliculus. And from the lateral geniculate nucleus, information travels to the visual cortex. One of the things you have to remember is that the lateral geniculate nucleus cells have center surround receptive fields. Okay, so in other words, it's like the little donut receptive fields that we talked about earlier, where there's the center, and then it's surrounded by another type of area. So there might be a center surround, for example, where it'll fire if it's stimulated in the middle but not in the outside area. That's a center surround receptive field. So the LGN cells seem to respond to center surround receptive fields, much like the retina does. Now, the major function of the LGN, of the lateral geniculate nucleus, is to control the flow of information as it's going from the retina to the visual cortex. So in a lot of ways, it's kind of like a filter of the information that's flowing from visual cortex, from the retina to the visual cortex. So the signals get received. Oh God, let's see. The signals get received. by the retina, and then flow into the brain, go to parts of the brain stem, the thalamus, and the cortex. The signals are ordered throughout the entire system, going through from the LGN to the visual cortex. The signals are organized by the eye and the receptor type and the type of environmental information. So which eye it comes from, right? Which receptor type it comes from, whether it's a rod or a cone, and the type of environmental information that we're looking at. Are we looking at a scene, a background scene? Are we looking at a face? What are we looking at? These are the types of organizational structure that's found in our visual system. The receptive fields, of course, have excitatory and inhibitory effects, if you remember, right? So, for example, the center surround receptive fields that Hubel and Wiesel were able to identify. Some of them were excitatory center, inhibitory surround. So if the receptive field received stimulation in the center, it would fire, and if it received stimulation, a light in the surrounding area, it wouldn't fire. And then the opposite was true: inhibitory center, excitatory surround, where if the receptive field received stimulation in the center, it wouldn't fire, but if it received stimulation in the surrounding area, it would fire. Okay. So all this information. One of the things that we did find is that the information that is processed by the LGN, there's also cortical information as well that flows from the LGN to the cortex, right? So the lateral geniculate nucleus doesn't just receive information from the eyes, it's also receiving information from the cortex. So there's this sort of feedback loop that goes from the cortex back to the LGN, probably to regulate expectations, right? What you're expecting to see. Hubel and Wiesel were able to identify neurons that fire to specific features of a stimulus. Okay, which is really interesting. This was the most exciting aspect of their research, right? As you get into the cortex, you start to find neurons that don't just fire to simple receptive fields, right? They actually fire to more complex features, to more complex things. There are cells in the cortex that are feature detectors, right? Hubel and Wiesel identified the simple cortical cell, the and what they would do is they would find a cell that would respond only to a bar on the screen that was oriented in a certain way. So, for example, let's say we had here the receptive field for a simple cortical cell, right? So this cell would fire when whenever the receptive field in the middle was stimulated, the center, but the surround wasn't, right? So they would put a bar, a bar in the receptive field, right? So they would have the cat watch this bar and make sure the bar lit up this part of the receptive field here. And when the bar was sitting in the middle in this excitatory area, the cell would fire like crazy. But if the bar was oriented such that part of it was in the positive area, which was right here, and parts of it were in the negative area, which is here and here, it would fire much less, as you can see here. And what they would do is they would orient the bar, so they would rotate it, right? They would simply rotate the bar, right? So this is the bar when it is perfectly vertical, right? And then they would orient it either positive 20 degrees or negative 20 degrees and simply measure how much the signal fell off as the bar kept oriented. And they were able to identify these receptive fields that were very specific to bars located in different parts of a visual field. One of the things they accidentally that they accidentally discovered was these cortical neurons that would only respond when a bar was moving in a particular direction, right? And the way they discovered this was by accident. They had an animal in the apparatus, and then they had found a particular cortical neuron that they were recording from. And while the animal was in the apparatus, they changed the slide that the animal was looking at. And as they moved, and you have to remember this was back in the day, right before we had digital video images, right? So they had to use projectors to project images on the screen for animals to look at. So they would get this old school projector here, and whoops, sorry about that. They would get this old school projector here, and take the slide out and pop it in. And they noticed whenever they put for this one cell that they were recording from, whenever they popped the slide in, the edge of the slide would go down, and this cell all of a sudden just went crazy. It fired like crazy, and that's how they accidentally discovered these feature detectors, these cortical cells that only respond when bars move in a particular direction. So, for example, the first one they discovered was the one they discovered accidentally was this one, right? So right here, for example, we would have it go down, right? And they found some cells that would do that, that would respond whenever this bar went from here to here. In this particular example, we're trying to figure out what a particular feature detector does, right? So we take the bar and we move it from here to here, and it doesn't do anything. And then we orient it slightly, about maybe 35 degrees or so, and then we have it go from here to here, and it fires a little bit. And then we orient it another direction and put it and move it, and it fires a lot. And then we orient it vertically and move it, and it doesn't fire at all. So we've identified that here, right here, is where the bar is where this particular cell fires the most, with bars that are only oriented at a particular that particular degree, and when it moves like that, right? So they were able to find hundreds and hundreds of these feature detectors that would fire only when bars were oriented and moved in a certain direction, right? Here's another one here, right, where if this bar was here, right, and you move it up, it fires a little bit. But if a little bit more of it is uncovered, right here, right, this area is uncovered, and then it moves up, it fires a lot, right, and it doesn't fire at all when it goes down, right. And then if we uncover it even more, right, and then move it up again, it only fires a little bit. So its optimal firing is right here, right? So these feature detectors are very specific, right? Notice the difference in where this edge is located, this edge is located, and this edge is located, right? Doesn't seem to be much of a difference, but it's this one right here where most of the firing occurs, right? So we put the edge here and then move it up like so, and it causes the most firing. So this is a listing of some of the types of cells that were found, right? So the optic nerve fiber, right? So this is the ganglion cell in the retina, and the ganglion cell in the retina is the cell that sends its axon through the optic nerve into the brain, right? So the optic nerve fiber has a center surround receptive field. So it's this round receptive field with the little donut hole in the middle, right, where it can be either excitatory outside, inhibitory inside, or vice versa, right? It responds best to small spots, but will also respond to other stimuli, okay? From the optic nerve fiber, it goes to the lateral geniculate nucleus. Also has center surround receptive fields, and they're very similar to the ones in the ganglion cell. From the lateral geniculate, we wire up with some simple cortical cells, right? And these are the ones that have those excitatory and inhibitory areas side by side, right? So with the three, the three squares stacked up next, or rectangles stacked up next to each other, where the middle one might be excitatory and the two on the side might be inhibitory. Uh, Hubel and Wiesel discovered that those respond to bars of different orientations. Complex cortical cells are the ones that respond to movement, right? So you orient a bar in a certain direction and then you move it, right? And then they also discovered those end stopped cortical cells, right, where they respond. It's the last example that I showed you. This one here. Whoops. Right here, these are the end stopped cortical cells, right? The ones where corners and angles are bars, are like put on top of each other and moved in certain orientations, and they fire only when the bars move in certain orientations. Um, if you keep stimulating one of these neurons, like the cortical cell or the end-stop neurons, they do start to get fatigued, right? They do start firing less as they get stimulated. So, of course, after a while, the neural firing rate will go down, and the neuron will fire less if the stimulus is presented again. Okay. When we say selective, we mean that only the neurons that respond to the specific stimulus adapt to that specific stimulus, right? So just because you have neurons that might respond to other stimuli, they're not the ones that get adapted. They're not the ones that get worn out or fatigued, right? It's only those neurons that are processing that stimulus that's getting presented over and over again that get fatigued.